Dear colleagues and friends, it's a pleasure for me to continue the discussion on further reference and considerations to the guidelines. I would clearly like to state here that the presentation will include some EMA, some PDCO, Pediatric Committee, but as well some personal perspectives. I'm Carine de Beaufort, I'm based in Luxembourg. I'm a PDCO-led delegate for this country, and anything I present is my personal view, and I've got almost no conflict of interest to declare, except that I do consider pediatric and adult type 1 diabetes a condition for which there is an unmet medical need of new drug development. So I'd just like to start with a short introduction about the pediatric regulation, this PDCO, which you will see coming back regularly. 2007, the objective was formulated to improve health of children in Europe by facilitating the development and availability of medicines for children aged 0 to 17 years. And this became a legal instrument with the aim really to get high quality research in the developments of medicine for children. And over time, a majority of medicine used in children who are authorized for this use, and to have high quality information about these medicines used in children. So the scope was the new products line extension of patent protected old products. And the procedure is the submission of a pediatric investigation plan, which needs to be associated with all these new products or line extensions. So the actors is clear, it's industry, PDCO, the PETCO, of course, as well, the EMA and the competent authorities. The incentive of this pediatric regulation was a 6 months extension of SPC, as well as free scientific advice and protocol assistance. And there has been quite some EU-funded research. Of course, every 10 years, you need to evaluate whether a regulation is what you expected or is doing what you expected to do. And here again, the focus we now really again established is to identify pediatric medical needs. And I do think the unmet need in type 1 diabetes exists. Strengthening as well the cooperation between decision makers and some practical aspects is timely completion of these pediatric investigation plans and improved handling, and of course always increased transparency around pediatric medicines. So just a quick word about the procedure, this pediatric investigation plan, the PIP. It is submitted that we call day of application 0, and during 2 discussion moments within the pediatric committee, there will be either an opinion when there are no questions, or there will be a request for modification. This request for modification comes at day 60, will then be transmitted to the applicant, who has time unlimited to come back. This is what we call the clock stop. Then when he resubmit, we call it day 61, but there can be several years in between. And then again, there is a limited time of 60 days, during which we again will have the discussion within the committee with the applicant to, in the end, arrive at a decision at day 120. Now back to the topic of today, the C-peptide and its role in remission. This is, of course, for many of you, a well-known slide coming from the paper of Insel in 2015, showing the 3 stages of type 1 diabetes. The discussion today is on the role of the preservation of beta cell secretory capacities in the stage 3, which is when we already have clinical symptoms of diabetes and a diagnosis of clinical diabetes, not the pre-symptomatic phase. Although I know that we are definitely very interested in that phase as well. So what is our objective? Our real objective is, of course, to obtain that remission in persons with an early onset type 1 diabetes in order to prevent as well chronic micro and macrovascular complications. But how to define the remission? I think if you go into literature, there will be quite a number of them. And I think it's important to consider that and say it's a limited or no exogenous insulin need to maintain normal glycemia. So in the EMA guidance, you'll find that the study design, the preferred study design, will be randomized and preferably, of course, double-blind and placebo-controlled in order to get very robust data. Inclusion, recent onset type 1 diabetes patients with a present or a still confirmed endogenous insulin secretion. And so the time limit will be quite dictated by the fact that there should be still endogenous insulin secretion present, although, of course, this can be discussed. And then a staggered age of inclusion, which will be based, of course, on the safety profile of the drug which is evaluated. So what would be really preservation of insulin secretory capacity? Clinical endpoint is, I think, those who have a complete insulin independence and an HbA1c within the normal range. But of course, we have got surrogate clinical endpoints, which already probably are pretty relevant. When you take the percentage of patients who have an HbA1c, we can discuss how high it should be or how low, and an insulin dose, which would be less than 0.5 units per kilo per day. Again, in the EMA guidance, you'll see that mode of action can be, is for the moment, mainly in the domain of the immunotherapy, and therefore safety data will have to be generated depending on the mode of action and safety profile, or expected safety profile. Duration should be at least 1 year for the primary outcome, but longer duration should be demonstrated after initiation of the treatment. So in the EMA guidance, again, you'll find potential co-primary endpoints. First of all, relevant for this discussion, the pancreatic beta cell function, which could be a change from baseline in C-peptide, for example, area under the curve, or, and if appropriately justified, the percentage of patients with a C-peptide increase above a clinically meaningful threshold following this physiological stimulus, where for the moment we will most often see the liquid mixed meal under of course, standardized conditions. Very important are the clinical co-primary endpoints, which still need to be there, because I don't think there is, well, anyway, let's not discuss this because that's the topic of this symposium, this workshop. HbA1c co-primary, there is still mentioned the frequency of hypoglycemic episodes. Here, I think we should only consider the severe ones, and we should discuss maybe in future whether this is still relevant, because current frequency of hypoglycemic events is going down, certainly in the first period after diagnosis. And of course, very important, the percentage of patients really without insulin. There are different secondary or exploratory endpoints which are considered: 24-hour glucose profile, this is still finger pricks, fasting and postprandial values, documentation, of course, on diabetes ketoacidosis, and the patient-reported outcome, which is a very important parameter to consider. I'm putting here, not yet in the EMA guidance as a secondary, but only as exploratory, the continuous glucose measurements, because I think this is something which has changed very importantly over the last 5 years. So some questions from my side, and this is a personal view. If we talk about pediatric beta cell function and change from baseline and the percentage of patients under a standardized condition, what do we consider as relevant, and what are standardized conditions? If you look at daily practice for those who are doing all those tests, 24 hours before test in a kit, there might be stress, there might be a difference in insulin need, there might be a difference in glycemic control, which might influence again the potential of the beta cell to secrete during the test. Can we consider that as the optimal way to collect these data, or is it the best we can get for the moment? Would fasting be better? Would a test be better? I think here we are working with the situation which is as good as it gets, but can we get better? And it would be really interesting to see, of course, requiring validation and qualification, but there we really would like to invite the different people to consider that. Would frequently dried blood tests measuring C-peptide at home be a more informative measure, because you can repeat it very easily, and people will already be doing this with still, I think, quite a lot who are doing at home the finger pricks. Should we consider 24-hour urine secretion and take that as a value? Because then at least we get, again, the home situation and 24-hour, which might be in some cases difficult because you don't know whether the collection is really 24 hours. Again, to be looked into. This is at the part of the beta cell function. As well, at the clinical side, with respect to outcome, we might need to consider in primary and in secondary other options. I think, as mentioned already, that the hypoglycemic episodes, very, very relevant, and certainly as patient-reported outcome, is still probably underestimating real hypos, or not really estimating hypos, because we don't know a lot what goes on when you're not measuring. And definitely you're not measuring continuously, except if you use continuous glucose monitoring. So here as well, I really would support further options to be submitted for qualification and validation, such as continuous glucose measurements, because I think there are more and more data popping up at at different parts, age groups and populations, which are extremely informative about what really is happening. Of course, then you need to consider again, if you take this as a surrogate validated or as a surrogate clinical endpoint, what measure, what metrics you're going to use, and which is going to be the most informative. Again, for the moment, future, but I really would like to take that on as food for thought. Further mentioned in the EMA guidance is the improvement of insulin sensitivity based on the fact that we get a modification in endogenous insulin secretion. Here again, not yet a validated surrogate marker for long-term improvement, could be potentially secondary and should not be underestimated, I think. And then the patient-reported outcome, several times mentioned already. The impact of treatment on quality of life is highly important, and I think it will be relevant to see whether we can get better standardized measures to evaluate this. So now a little bit still on the PIP, what have we seen in these pediatric investigation plans as endpoints over the last 15 years almost? We've had the co-primary classic, as already discussed before. We've had some isolated primaries, with one which was only looking at the area under the curve within the first 12 months, and we've had one where there was a composite marker, which nowadays is less applied because there are different data popping up in literature. That's this insulin dose adjusted A1C, and where was being looked at the change over time. Now these are what we've seen, but as mentioned, some are already older studies. Secondary outcomes which have been evaluated in the PIPs are listed on this slide. Again, I think some have been already discussed and are validated. Others are still exploratory or not yet belong to the validated measures, but very informative. I think in patients, we always need to be careful as well. If we talk, for example, about insulin requirement, it's going to be not always that easy if we haven't got documented downloads from pumps on to decide what really has been, or what has been said, or what is expected to have been used. I think in that sense, if you look in pediatric diabetes, there are more uncertainties than certainties about what is and can be reported. So I really would be very much in favor of CGM and downloads to be discussed and be looked upon as potential markers which should get qualification evaluation. So in summary, co-primary, we still need the clinical endpoints, HbA1c, insulin requirement administration, and pancreatic beta cell function evaluation, the C-peptide is still area favorable, would be the area under the curve during the as much standardized as possible mixed meal test. But I would really endorse to see whether we can get a validation qualification for continuous glucose monitoring, dried blood spots, C-peptide measurements, and potential insulin sensitivity measures. And just food for thought as well, I think we are moving forward in a time where heterogeneity of the disease is becoming more and more clear, and where we might consider new approaches in order to evaluate our work. Thank you very much for your attention, and looking forward to further discussion.